Hi, this is Nick Cap, and this is your Skyline Microbiology Laboratory Minute. Today what I want to do is to talk about a little bit about the techniques that we will be using in the coming weeks. We will be covering Chapter 9, looking at dilutions, Chapter 11, looking at kind of exoenzymes, those enzymes that are put outside the cell, and then number 12, looking a little bit at carbohydrate metabolism. What I basically wanted to give you is the basic ideas of what techniques that you will be using. So in our serial dilution, or dilutions, we will be doing a serial dilution in order to counter bacteria. And so if we look at this sheet right here, we will be doing serial dilutions. What we're going to do is we're going to take a culture of bacteria, we're going to take out 1 ml of that bacteria, and we're going to put it into 99 ml of sterile water. And so here's the water that we have. And that is essentially a 1:100 dilution. We will do that dilution one more time. Again, take that 1 ml, and we will put it into 99 ml of water, and then that is a 1:100 dilution. But in total, it's a 1 to 10,000 dilution. And so if we started out with 10 cells, if we started out with 10,000 cells per milliliter, again, in this one here, we would have 1,000 cells. And then a 1:100 dilution, we would have 10 cells. Okay. So let's go ahead and see what that looks like a little bit. That's the calculations that we will be doing, and that's the calculations that we've done in class. So you would have to have your dilutants here. We are going to use a serological pipette, and a serological pipette, basically the whole volume goes down to the very tip, so you don't leave any in this pipette. This pipette is sterile, and so what you're going to be doing is kind of opening up the pipette from the opposite end. Try not to touch the pipette. You will use a pipette aid to put your pipette in here, or slowly get this. This goes in the garbage, wherever the garbage is. And then what you're going to do is you're going to, again, go into a tube, and you're going to take out, in this case here, that's 1 ml. If you can see it, if you can focus in on the top. Put my tube back. And then this 1 ml of media, you're going to go into your, into your 1 ml tube. If you see the color in there, and you can go ahead and mix up the tube. What you will then do, and you can actually see this one here is slightly pink. You'll get rid of your tube, and this will go into biohazardous waste. We will have that for you. Now, I'm going to show you a little bit different technique. We actually have another piece of equipment that we can use to move 1 ml, and this is called a pipetter. And you can take this pipetter, and you can get a sterile pipette tip. It's set to 1 ml. I would press it down until I first feel resistance. Try not to touch the sides. Go in, take out a sample. Okay. I would then put it into the next tube. Okay, and then you're done, and you would dispose of this in the right way. And again, this is a 1 to 10,000 dilution, and as you can see, it goes from being colored to having a light red color to being basically clear, because we've diluted out so many times. The next I want you to show is basically how will we count this stuff? What will we do? And again, on this guy here, I'll use some of the plates that we have. You're going to use a turntable, and it spins like this. You're going to use a Petri plate that's on there like this. And then what you will do is, I'm going to use this. You're going to get your sample. Using a sterile tip, you're going to get your sample, and you would take up between 0.1 ml and 0.2 ml, and you would put them, go ahead and seal this up. You would go ahead and put them on your plate. Okay. And this tip would be disposed of properly. I'm not doing that now. And then you can either use a disposable plastic spreader bar, or in this case here, I have a spreader bar that's in some alcohol, and this will allow us to use it. And it's a 70% alcohol that actually sterilizes it. Now I need to get rid of my alcohol in order so I don't kill my culture. So what I will do is kind of burn off that alcohol. If you can see, I'm burning off the alcohol. I don't want to put it on there too much because I don't want my spreader bar to become hot. The reason why I'm burning it is to get rid of the alcohol. And then what I can do is I can touch either the top of the plate or on the side, and then I will use the turntable and just spread it, and I will move it around, and I move the bacteria all evenly on the plate. When you're done with this will go back in the alcohol, and that will kill the bacteria. You're going to let these plates dry for about 5 minutes. Then you can invert the plate and safely put them in the incubator. Okay? That was Chapter 9, looking at our dilutions and spreading. This is Nick Cap, and that was your Microbial Lab Minute.